Hi class! So, this week we will learn a new chapter, which is chapter 6, about plasma membrane and transport molecules, in which in this chapter you can learn about the two types of plasma membrane models, the transport of small and also large molecules through the plasma membranes. So, what is plasma membrane? Plasma membrane, also known as the cell membrane, it is a thin semi-permeable membrane that surrounds the cytoplasm or the content inside the cell. So the plasma membrane functions as the boundary that helps to separate the living cells from its outside surrounding. And also, plasma membrane exhibits selective permeability, meaning to say that it only allows some substances to cross it more easily than others. And also, plasma membrane help in recognition and also communication between cell and tissues. So this is how the plasma membrane looks like. However, there are two types of models that being proposed about plasma membrane, in which in 1935, Hugh Davidson and James Danieli proposed a sandwich model, in which in sandwich model, the phospholipid bilayer lies between two layers of globular protein. Later, the studies found problem with this model because the placement of positions of the membrane protein is wrong, which have hydrophobic and also hydrophilic region. As you can see over here, the phospholipid bilayers lie between the layer of the protein. Then later on, in 1972, Singer and Nicholson proposed the fluid mosaic model, in which these models are being used until now, in which that membrane is a mosaic of protein that disperse within the bilayer, either on the embedded or on the surface of the phospholipid bilayer, with only the hydrophilic regions, which is the parts that likes water, exposed to the water, and the hydrophobic region, which is the one that does not like the water, not exposed to water. So as you can see here, some pass through across the plasma membrane, across the phospholipid layer. Some proteins are only embedded at the phospholipid layer. This is a fluid mosaic model. So this is how it looks like, in which we can see the hydrophilic region over here being exposed to the cytoplasm and also to the surrounding. Meanwhile, the hydrophobic region embedded in the hydrophobic part of the phospholipid bilayer. So the freeze fracture, there is a preparation technique which is called as a freeze fracture that helps to split the membrane along the middle of the phospholipid layer, in which this freeze fracture study of the plasma membrane supports the fluid mosaic model. As you can see over here, the cells is being freeze and then being cut using a knife to separate the two layer of the plasma membrane. And as you can see here, this is known as the extracellular part, in which inside the extracellular part it is much softer compared to the inside of cytoplasmic layer that showing lots of the proteins. So next, we're gonna look on the components of the plasma membrane, in which first the phospholipid bilayer, the membrane proteins, membrane carbohydrates, and also the cholesterol. So for the phospholipid bilayer, phospholipids are the most abundant or the type of lipid that make up the plasma membrane. So phospholipids are amphipathic molecules. What does it mean by amphipathics? Amphipathics meaning to say that molecules contain both hydrophobic and also hydrophilic region. As you can see here, this made up from the phospholipid, and it known as a B layer because they have two layers of phospholipid, first layer and the second layer, and also it do have a hydrophilic area region on the head and also the hydrophobic region on the tail. So phospholipid in the plasma membrane can move within the bilayer, and most of the lipid and some protein they are moved in a horizontal direction and rarely does a model flip-flop across the membrane. So let's look here. Okay, usually the phospholipid will move in a horizontal direction, either to the right or to the left, and they are rarely moving across each other, which is known as a flip-flop movement over here. So as the temperature cools, the membrane will switch from a fluid state to a solid state. Okay, the temperature at which a membrane solidifies, it depends on the type of the lipids. So the membrane which is rich in unsaturated fatty acid, remember unsaturated fatty acid are liquid at room temperature, are more fluid than those plasma membrane that saturated with fat, that rich with saturated fatty acid. And membranes must be fluid to work properly, and they are usually about as fluid as salad salt, so they need lots of unsaturated fatty acid to make it more fluid. Okay, so as you can see over here, the plasma membrane that rich with the unsaturated fatty acids are more fluid compared to the one that rich with saturated fatty acid. This one is more viscous compared to the unsaturated fatty acid one. Next is the membrane protein. So different protein often being grouped together, embedded in the fluid matrix of the lipid layer, and proteins will determine most of the membrane specific functions. So there are three types of membrane protein that can be found in plasma membrane. The first one is peripheral protein that bound on the surface of the membrane. Next is second integral protein that will penetrate across the hydrophobic region, in which the integral protein that across along through the plasma membrane known as a transmembrane protein. So the hydrophobic regions of an integral protein consists of one or more stretches of unpolar amino acids, which is often called into alpha helixes, as you can see over here. Okay. Excuse me. So as you can see here, the peripheral protein stays on the surface of the plasma membrane. The integral proteins are the protein that integrated into the membrane. Meanwhile, the integral protein that will across the plasma membrane known as a transmembrane protein. So 6 major functions of membrane proteins. The first one is for transport, in which it will transport molecules such as sodium and potassium, and it also acts as a biocatalyst or the enzyme, and it also important for signals transduction, also for cell recognition between the two cells, for intercellular joining, and also for the attachment of the cytoskeleton, and also the extracellular matrix, which is the ACM. Next, the component of the plasma membrane that we need to know is the membrane carbohydrates. So there are two types of membrane carbohydrate. First is glycolipids. Glycolipids are the membrane carbohydrate that is covalently bonded to lipids. Meanwhile, glycoprotein is a membrane carbohydrate that covalently bonded with the protein, in which the carbohydrate on the external sides of the plasma membrane vary among the species, individuals, and even cell types in an individual cells. So most of the carbohydrate proteins function in cell recognition, in which the cell will recognize each other by binding to surface molecules, often containing the carbohydrates, on the extracellular surface of the plasma membrane, as you can see here. This one is known as a glycoprotein. As you can see here, the glycoprotein are bonded, the carbohydrates are bonded with the protein. Meanwhile, the glycolipids, the carbohydrate, are bonded with the phospholipid part. So it helps to cell recognition, as you can see here. So next, the component that also important in plasma membranes is the cholesterol, in which the steroid cholesterol has different effect of plasma membrane fluidity at different temperature. In warm temperature, cholesterol will restrain the movement of the phospholipid, and at cool temperature, it will maintain the fluidity by preventing the tight packing of the phospholipid bilayer. So as you can see here, the cholesterol are between the phospholipids. So how about the permeability of the phospholipid bilayer or the plasma membrane? The cell, remember, a cell must exchange material with its surrounding, in which this process is controlled by the plasma membrane. Plasma membrane are selectively permeable because of its structure. This enables the regulations of the cell's molecular traffic. So the hydrophobic molecules, such as carbohydrates, can dissolve in the lipid bilayer and pass through the plasma membrane rapidly and easily. Meanwhile, the hydrophilic molecules, such as polar molecules, such as the sugar and also water, do not cross the membrane easily due to the hydrophobic region of the plasma membrane. Okay, so as you can see here, the small and also hydrophobic and also small and hydrophilic molecules can pass through easily the plasma membrane. Meanwhile, large or charged substances cannot pass through the plasma membrane easily. So remember, one of the components of the plasma membrane is transport protein. Okay, so transport protein allow the passage of hydrophilic substances across the membrane. And some proteins are called as channel protein, in which channel protein have a hydrophilic channels that will help to transport molecules or ion to pass through the plasma membrane. And channel protein that facilitate the passage of water is known as aquaporins. Meanwhile, carrier protein will bind to molecules and it will change shape to shuttle them across the plasma membrane, such as sodium and potassium pump. So a transport protein is very specific for a substance that it moves. So as you can see here, this is a channel protein and also a carrier protein. A channel protein such as the aquaporin, meanwhile carrier proteins such as sodium and potassium pump. So next, we're going to look on the transports inside the plasma membrane. So transports of small molecules involving passive and also active transport. So passive transport is actually a diffusion or a transport of substances across a membrane with no energy investment, meaning to say there is no ATP is being used and the molecule is transferred from high concentration region to low concentration region. Meanwhile, active transport is the movement of substances that use energy because the movement of the substances is against its own gradient, meaning to say from low concentration gradient to high concentration gradient. So, passive transport, there are three types of passive transport: simple diffusion, osmosis, and also facilitated diffusion. So, what is simple diffusion? Simple diffusion is a diffusion that involves the movement of molecules across the plasma membrane from high concentration to low concentration gradient. So, although each molecule moves very random, diffusions of populations of molecules may be directional. So, at dynamic equilibrium, as many molecules cross the membrane in one direction as in one another. So, the substances will diffuse down because their concentration gradient from high to low region. So as you can see here, this is the examples of the diffusion of one solute in which it will move from high concentration gradient to low concentration gradient until it reach equilibrium. Meanwhile, this is the examples of diffusions of two solutes in which both solutes will movement until it reach equilibrium. The second type of passive transport is osmosis. Osmosis is diffusion that involving water, diffusions of the water across a selectively permeable membrane from region of lower solute concentration to region of higher solute concentration until the solute concentration is equal in both sides. As you can see here, the solute, which is the sugar, in the first part is very low compared to the sugar or the solute in the second part of the water. Meaning to say, this part is more saturated or less concentrated compared to the second part. So what will happen? The water from the lower concentration of solute will move to the region with high concentrations of solute. Therefore, both of these parts will reach the same concentration level between each other. Excuse me. And osmosis involving the tonicity, in which tonicity is the ability of surrounding solution to cause a cell to gain or lose water. So there are three types of surrounding solutions. We have isotonic solution, hypertonic, and also hypotonic. Isotonic is when the solute concentration is the same between the outside and inside the cell, meaning to say there is no net movement across the plasma membrane. For hypertonic solution, the solute concentration inside the cell is greater compared to outside the cell is greater compared to the inside the cell. Therefore, water will move out from the cell from lower concentrations of the solute to the higher concentration of solute region. Therefore, the hypertonic the cell will lose water. Meanwhile, for hypotonic, the solute concentration outside the cell is less compared to the inside the cell. So what will happen? The cell will move from low concentrations of solute outside the cell to the region with high concentrations of solute, which is inside the cell. So the cells will gain water from the surrounding. So as you can see here, for hypotonic, the hypotonic, the water will move into inside. It will move into the cell, so the cell will gain water due to the high concentrations of solute inside the cell. Isotonic, both part have same concentrations of solute. For hypertonic, the concentrations of the solute at the outside of the cell is higher compared to the inside of the cell. Therefore, the water inside the cell will move out and the cell will lose water. So as you can see here, the effect of the solution, three types of solutions on the animal cells and also plant cells. So for hypotonic, the water moves into the cells. Therefore, for animal cell, it will cause the cell to expand and finally it will burst. And this condition is known as a hemolysis if it involves a red blood cell, because there is a lot of water going inside the cells. For the plant cells, when the water enter the plant cell, the cell will become very turbid. For isotonic, no net movement between inside and outside of the cells. Therefore, the animal cells remain the same, and also the plant cell also remain normal. If you put the animal cell and plant cell inside hypertonic solution, so what will happen? Animal cell, it will shrink because the cells lose lots of water. Meanwhile, for plant cells, what will happen? The cells will become very flaccid as the water move out from the cells. So in animal cells, hypertonic and hypotonic environment create osmotic problems for organisms, such as the osmoregulation. Osmoregulation is the control of solute concentration and water balance, and this osmoregulation is necessary for adaptation for life in such environment. For example, paramecium. Paramecium, which is hypertonic to its pond water environment, they have a contractile vacuole that will help to pump out the excess water that goes into the paramecium body. Meanwhile, in plant cell, the osmosis will help the osmosis, okay, will being helped by the cell walls to maintain the water balance, in which in hypotonic cells, a solution, the cell will become turgid. Meanwhile, if you put them in the isotonic reactions, there is no net movement inside the cell and they may wilt. Meanwhile, in a hypertonic environment, the cell will lose water and it will cause the plasmolysis to occur, and the membrane will pull away from the wall as a usually lethal effect to the cells. So this one is referred to the benefits to human. It depends on the isotonic. For isotonics, it will quickly replace the fluid that being lost by the sweating, and also it will supply a boost of a carbohydrate. For this is very suitable for average athlete and also athlete with team sport. Examples of isotonic drinks are Powerade, Gatorade, and also 100 plus. Meanwhile, for hypertonic drink, the benefit is for supplement for your daily carbohydrate intake because it contains high level of sugar. So athletes drink it after exercise to help top up your body muscle glycogen stores, and also for a long running distance running athletes such as the Red Bull, Cola, and also fruit juice. Meanwhile, for hypotonic, which is quickly replace the fluid loss through sweating and it's very low in carbohydrate. This one is very suitable for jockeys and also gymnasts. This one is referred to the normal mineral water. So the the third type of passive transport other than osmosis and also simple diffusion is facilitated diffusion. In facilitated diffusion, the movement of molecule across the plasma membrane with the help of transport protein from high concentration gradient to low concentration gradient. Okay, it seems with the simple division, however, it does need the help of transport protein. So there are two types of transport protein that being discussed before, which is channel protein and also carrier protein. So for channel protein, it will provide a corridor or a way that allow specific molecules or ion to cross the membrane. It includes aquaporin that for facilitated diffusion of water, and also ion channel that open and close in response to stimulus or known as scatter channel. And for carrier protein, it undergoes subtle change in shape that translocate the solute binding site across the membrane. And some diseases are caused by malfunction in specific transport system. For example, the kidney disease cysturia. This is how the carrier protein and the channel protein look like. So next we're going to look on the active transport. Active transport is the movement of ion or molecule across the membrane from low concentration gradient to high concentration gradient with the help of energy and also protein. Okay, and it performed by specific protein that embedded in the membrane, such as sodium and potassium pump. As you can see over here, the transport of sodium and potassium pump needs energy to be carried on. Ion that pump will maintain the plasma membrane potential, in which it involves the voltage across the membrane. The voltage is created by differences in the distributions of positive and also negative ion across the membrane. So two combined forces that called as the electrochemical gradient will drive the diffusions of ion across a membrane. The first one is chemical force, and in which it depends on the ion concentration gradient, and also an electrical force, in which it depends on the effect of the membrane potential on the ion movement. So an electrogenic pump is a transport protein that generates voltage across a membrane, and such as sodium and potassium pump in animal cell, and also proton pump mainly can be found in plant, fungi, and also bacteria. Okay, as you can see here. Meanwhile, a cold transport occur when the active transport of solute will indirectly drive the transport of the nearby of other solutes, as you can see over here. The transport of through the proton pumps, it will drive the transport of the other solute, such as the sucrose co-transporter. So remember three types of passive transport: simple diffusion, facilitated diffusions, and osmosis, and also facilitated diffusion. Meanwhile, for active transport that need energy from low concentration gradient to high concentration gradient. So next, we're gonna look on the transport of large molecule, which is exocytosis and endocytosis. So large molecule transport across the plasma membrane occur by exo, and endo. So large molecules such as polysaccharide and also protein cross the membrane in bulk via vesicle. So bulk transport require energy, meaning to say it is under the active transport. So for exocytosis, what will happen? The transport vesicle or secretory vesicle will migrate to the membrane. Okay, as you can see over here, combine with the membrane, and then it will, as you can see over here, and it will release its content. So many secretory cells use exocytosis to export the product. As you can see here, the transport vesicle or secretory vesicle will fuse together with the plasma membrane and then release their content. Meanwhile, for endocytosis, the cell takes in micromolecule by forming vesicle from the plasma membrane. So it is a reverse of exocytosis, and it involving three different proteins, which is different proteins, and they are three types of endocytosis, which is known as phagocytosis, cellular eating, pinocytosis, cellular drinking, and receptor-mediated endocytosis. So as you can see here, for endocytosis, first what will happen, there will invagination will occur in which the molecules from the surrounding will goes to the plasma membrane, and then what will happen, the plasma membrane will form a pouch here, and then it will form a vesicle to import the molecules from the outsides of the cell into the cells. So three types of endocytosis just now, as I mentioned: phagocytosis, the cellular eating; pinocytosis, cellular drinking; and receptor-mediated endocytosis. So phagocytosis or cellular eating is a process where the cell will engulf a particle in a vacuole, and the vacuole will fuse with lysosome to digest the particle that being engulfed. For pinocytosis or cellular drinking is a process where molecules are taken up when extracellular fluid is gulped by the organism, in which it will swallow very rapidly into tiny vesicles. Meanwhile, receptor-mediated endocytosis is the process where the ligand will bind to the receptor and it will trigger the vesicle formation from the plasma membrane. A ligand is actually referred to any molecules that bind specifically to a receptor site of another molecules. So as you can see here, they will engulf, phagocytosis engulf the solid from the outside and then form a vesicle, and then it will combine with the lysosome to digest the food molecules. For pinocytosis, okay, they will swallow the liquid very rapidly, and also for the receptor-mediated endocytosis, what will happen? The ligand here, okay, which is in purple in color, will bind to the receptor and it will trigger the plasma membrane to form a vesicle, as you can see here. So this is the difference between endo and exocytosis. Endo molecules are taken into the cells. Meanwhile, exo, the contents or the molecule are being exported to the outside surrounding of the cells. So that's all for today, class. Thank you.